The presence of seminal stain is very important in crimes involving a sexual offense. Semen detection is an important factor in forensic investigation to confirm sexual assault in cases of rape.
Many sexual offense cases involving seminal fluid of various types were submitted to Forensic Science Laboratory for analysis and detection.
Once this is determined, it is important for the DNA analyst to determine if sperm is present in the fluid because it contains a great deal of DNA, which could help to identify a suspect.
Once it has been determined that sperm is present, the analyst must determine which item of evidence or swab sample has the most sperm so that getting a DNA profile is most likely.
Sperm cells, enzymes, sugars, minerals, organic chemicals, and vitamins. What are the glands that contribute secretions to the seminal fluid? There are 4 glands. First, testis, seminal vesicle, prostate, and bulboretal gland. Semen can be one of the forensic evidence. Why? Because semen is a thick, yellowish-white, glary or pearlescent, and secretion having a characteristic odor known as seminal odor. It is also called seminal fluid, fluid that is ejaculated from the male reproductive tract and contains sperm cells. Did you know that stains will usually be found on the clothing of the victims or the perpetrator? Particularly the undergarments stains may also be found on bedding, mattresses, car seat, and etc. Clothing and articles suspected to contain seminal stains should be seized quickly as possible because an attempt may be made to remove the traces by washing. Often, seminal stains are readily visible on fabric because they exhibit a stiff, crusty appearance. The female victim of an assault may have a considerable quantity of semen in or around the vagina. In such cases, vaginal swabs should be obtained through the medical examiner. Controlled samples should be collected from both the suspect and the victim. What is the importance of semen as forensic evidence? First, semen analysis is a crucial laboratory test that should be evaluated in the same way as any other diagnostic test. Second, semen analysis is very important in assault cases as it is very useful for the detection of semen in clothes. Third, isolation and identification of seminal stains found on various suspected areas such as clothes, bed sheets, pillows, blanket, and etc. Fourth, analysis of various proteins present in semen or seminal stain. Fifth, seminal stains are examined generally in rape cases. And lastly, detection of sperm cells in the vagina and other body parts. The downside of the previous method in analyzing semen stains, establishing the fact of sexual activity is particularly crucial among the forensic medical examinations conducted in sexual offences. However, techniques for recognizing and analyzing semen stains have not yet been sufficiently developed, necessitating the creation of new, efficient and cost-effective techniques for detecting the presence of sperm on physical evidence. Is there any new developed method to overcome this problem? Yes, a new approach for obtaining precipitating serums specifically reacting to human seminal fluid intended for use in forensic laboratories has been developed based on recent scientific research that was conducted on 8th of February 2023. This method involves hyperimmunized animal producers.
New approach for obtaining specific hyperimmune serum for the detection of seminal fluid on the physical evidence by serological method was based on this article, where it used hyperimmunization of animal producer as a serum. So, what is hyperimmune serum? Hyperimmune serum, also known as antiserum, are specialized serum that contain high level of antibodies specific to a particular antigen. They are typically generated by immunizing animals such as rabbit or horses with the target antigen to obtain strong immune response. This antibody can then be collected and used for various diagnostic and research purposes. In the context of detecting seminal fluid on the physical evidence, hyperimmunoserum can be employed as the presumptive test in the serological method to identify specific protein or antigen present in the semen. By targeting unique protein found in the semen, serological tests can help to determine the presence of seminal fluid in the item like clothing, bedding, or other physical evidence. Hyperimmunoserum involves several processes to detect seminal fluid using serological method. We want to focus more on the third step, but before that, let's go one by one for each processes. The first step is collection of physical evidence. The physical evidence, such as clothing or bedding, that's suspected of containing semen, is collected and properly preserved to maintain the integrity of the sample. Second step is extraction of seminal fluid. The semen is extracted from the physical evidence using appropriate technique. This may involve cutting out a small portion of the stain area or using swab to collect the sample. The third step is preparation of the hyperimmunoserum. Based on this article, they use 40 chinchilla rabbit between 2.5 to 3 kg as producer of the serum. The animal that passes the veterinary examination were kept in quarantine for 45 days. Then, the antigen was put in several places near the lymph node to stimulate the production of antibody. After making sure that these antibody were absent in the blood of the producer was subcutaneously injected with the antigen in the form of human seminal fluid. Next, 4 groups of 10 rabbit were formed. Each group's animal were immunized differently. First group rabbit were immunized with 2 ml per kg of antigen every 15 days. Same procedure for the second group, but additional antibiotic was used. Furthermore, antigen was given to group 3 every 5 days in increasing doses of 1, 1.5, 2 and 2.5 ml per kg. With similar procedure, antibiotics are added to the fourth group. The blood serum of immunized rabbit was taken to check for the presence of human seminal fluid antigen every 5 days. After being immunized 4 times, blood was taken from the heart cavity 7 days later. Separated rabbit serum were examined for specificity using animal and human antigen in the form of blood plasma at dilution of 1 to 100. The last step is serological testing. The purified hyperimmunoserum are then used in serological tests to detect the presence of seminal fluid antigen on the collected physical evidence. Various serological methods can be employed, and one commonly used method is enzyme-linked immunosorbent assay, ELISA, where an enzyme-linked secondary antibody is added, which produces a color change when the enzyme reacts with a substrate. The intensity of the color change indicates the presence and quantity of the seminal fluid antigen in the sample.
In research result, the study of the titer of antibodies in the blood serum showed that all 40 rabbits formed specific antibodies to human seminal fluid after immunization. Post-injection abscesses were observed in 8 animals of group 1 and 7 of group 3, while the animals of the second and the fourth groups who received antibiotics had no abscesses. The antibody titer did not exceed 1:100 in rabbits with post-injection abscess. The intensity of antibody formation in group 1 was relatively low, with only 2 animals of this group having an antibody titer higher than 1:500. The intensity of antibody formation in group 3 was higher than in group 1, with 5 rabbits having an antibody titer exceeding 1:500 at the end of the immunization. The second group of animals immunized with an interval of 15 days formed a titer of 1:500, while 8 animals had a titer above 1:500. Serums obtained from rabbits were tested for specificity by antigens from cattle, poultry, and humans in a 1:100 dilution using the Kistovich methods. In the study, precipitation rings and bands were not formed within an hour. Serums with a titer above 1:500 were used to determine seminal fluid on physical evidence, and positive results were obtained in all cases.
In conclusion, the ability of forensic biology laboratories to examine physical evidence has been improved by the use of serological research methods in forensic medicine, particularly the precipitation reaction, which uses precipitating serum to detect seminal fluid. Use of antibacterial drugs in conjunction with antigens during animal immunization allows for the achievement of reasonably high antibody titers in the animal serum
as well as the prevention of abscesses from forming.
It is possible to create precipitating serums with a titer of at least 1 to the ratio of 500 that selectively react to human seminal fluid, using the suggested method of hyperimmunizing animals. In turn, this enables the serological technique to detect the presence of human sperm fluid.